Bismillahirrahmanirrahim. We start another paper 2, and I hope this is going to be helpful for your upcoming exam, which is just in a few days. Now, as you all know, section A is only one section in this paper, and you have to answer all the questions. Now, in question 1A, figure 1.1 is a diagram of a human chromosome at a stage in mitosis. You see, what you have to understand is that this is one chromosome actually. And then what has happened is DNA replication has taken place, and this is an exact copy of it. I'm just coloring it differently. And what it has is that it has this area here, and this area here is called the centromere, to which the spindles are going to get attached. So figure 1.1 is a diagram of a human chromosome. So now, as they are now copies, now these will be called chromatids. But when they are separated, then if this one is separated, then this will be called a chromosome. We usually do not like mixing up these names, but sometimes it's allowed. The paragraph describes the structure of the chromosome shown in figure 1.1. Complete the paragraph using the most appropriate term in each space provided. The chromosome shown in figure 1.1 has two genetically identical sister chromatids joined at a centromere. The chromosome is composed of two DNA molecules, each wrapped around proteins known as histone protein. So that was a very easy 3 marks. I'm sure everybody knew that. Then it said state one stage during mitosis. State one stage during mitosis when the chromosome would appear as shown in this, and that is prophase, or if you had said metaphase. Then suggest the role of ATP in the process of mitosis. Now ATP has a universal energy currency, and suggest the role of ATP in the process of mitosis. Number one, it provides energy. Number two, movement of the centrioles to the poles. All the activity which is taking place which requires movement or formation of something, like number one provides energy. Then we said movement of centrioles to poles. Then I said spindle formation. Then movement of chromosomes to the spindle equator. Then separation of the chromatids during anaphase. So all these are very active processes and need energy. So if you need energy, well, ATP is the universal energy currency that is going to provide you the ATP. So without ATP, nothing can happen. And it is the entire your body temperature is maintained because of ATP. So basically all reactions have to take place which require ATP, and ATP is the universal, so provides energy. And coming to the B part of the question, prokaryotes are bacteria. So prokaryotes divide by a process known as binary fission. Here shows figure 1.2 some of the stages in binary fission. This is the DNA, then this is the cell surface membrane and the cell wall. Now you can see it has become, now if you look at this, if you compare this diagram with this diagram, this cell has entirely become a little elongated, and the DNA now is looking a little more complex. We still don't see it. And then we see this sort of a division occurring, and then this dividing into two prokaryotes. Now we don't study binary fission, so they've shown you a diagram of it, and they've shown you how the cell is elongating first, and then it is sort of in the center there's a cleavage, and then it is dividing into two, and we have now this one is finished. There is no more this one left. Now we just have this and this. Now it says with reference to figure 1.2, identify two events that occur during binary fission but that do not occur during mitosis. Now I'm going to revise mitosis first, and then I'm going to come back to this mark scheme. Now if you look at the cell cycle, which I have shown you a diagram of the cell cycle, so this stage is the entire stage is interphase, and in interphase DNA, in S phase of interphase, DNA replication takes place. So DNA replication takes place during S phase of interphase, and DNA, please remember, in human cells is linear and is chromosomes. And then which part is mitosis? Mitosis is only the part which is prophase, metaphase, anaphase, and telophase. So, I mean, my mnemonic, which I always say, is I Now let me just correct it. I is for interphase, and then I have a purple mat, IP mat. So this is the only part which is mitosis, and then of course the other part, which is cytokinesis, also does not take place. And the cytokinesis is a separate part, and this is not part of mitosis. So just a quick revision for you. So interphase was where DNA replication, or S phase of interphase was where DNA replication was taking place. This is S phase of interphase. So if you have really understood this part, then you can do that part of the question. This question again. With reference to figure 1.2, identify two events that occur during binary fission do not occur in mitosis in human cells. So DNA replication does not take place in mitosis. Cells do not get longer. So now, whenever there is something in which there is a comparison, you must give me what you're talking about. Are you talking about binary fission, or are you talking about mitosis? If you do not write anything, now you can see here I've written binary fission. Why? Because if you do not write anything, we will assume it is the first one which you are comparing. So the first one is binary fission in the question. So I'm going to assume that you're talking about binary fission. But if you said DNA replication takes place in mitosis and you didn't say anything, I would assume that if mitosis had been written first, then what are we talking about? So binary fission is written first and then mitosis. So please, you must give the heading. We're talking about binary fission, or are you talking about mitosis? So am I talking about binary fission? Yes, I wrote that and I said yes, in binary fission DNA replication takes place, but not in mitosis. In mitosis it doesn't take place. Cells get longer here, not in mitosis. Cell wall formation, not in mitosis in human cell. There is no cell wall in humans. And here there is cytokinesis. Here there is this cytokinesis. This goes separately, this goes separately. So this is cytokinesis. While mitosis, it was no cytokinesis. I just revised that with you. Cytokinesis is a separate part. It wasn't part of mitosis. And the next one is question 2. Question 2 is figure 2.1 shows a molecular structure of a triglyceride molecule. Then it said draw a circle around an ester bond shown in figure 2.1. I've just drawn that. You only had to draw one an ester bond, but there are three ester bonds. Then it says name the type of reaction used to produce triglyceride from its components. And the type of reaction is condensation, and the number of water molecules produced will be 3. Naturally, because I've just circled the 3 bonds. So the glycerol molecule, this is the glycerol molecule, and it gets attached to 3 fatty acid chains. And so 3 waters will exit here, here, and here. So 3 water molecules will exit, and that is why we said 3 here. The part B of the question, lipases are enzymes that digest triglycerides in the lumen of the human intestine. These enzymes are released by exocytosis from intestinal epithelial cells. Underline all the terms from the list that are used to describe these lipases. Now, you know lipases is an enzyme. Enzymes are proteins. So it's a macromolecule. 1 mark. Underline. Underline. An extracellular enzyme because they work outside. They're produced inside the wall of the intestine, but they work in the lumen of the intestine. So it is an extracellular enzyme. It's not a fibrous protein. It's not a polysaccharide. And I'm sure everybody knew that. And this was going to get you 1 marks if this was underlined and this was underlined. Scientists have found that treating milk with lipase can improve its taste. The scientists carried out an experiment to determine the effect of lipase activity on the triglyceride found in milk. Lipase was immobilized in alginate beads. The pH of a known volume of milk was adjusted to pH 8 by adding an alkali. The beads were then mixed with the milk in a beaker. The pH of the reaction mixture was recorded over a period of 24 hours. The results are shown in figure 2.2. I often say is you need to look at the graph and you need to talk about the graph and you need to be sort of explaining the graph to an imaginary person sitting across you. And we look at the x-axis and it is time in hours, and we've done it in up to 30 hours, but of course we did it for 24 hours as we said in the question. And then you can see here pH of the reaction mixture, now how it has decreased. It was 8 here and then it has decreased and it's really coming down very fast and it's come to 6.6 in the first 2 hours, and then it has gradually decreased, and then it is leveling out. When it levels out, we say plateau. And I hope you all know this word, what plateau means. So from 6.6 went down to 6.4, 6.2, and the last is it's something like 6 point something here. It's even less than, it's nearly 6.1. So what we have to understand is that how it has decreased from 8 to 6.1 in about 24 hours. Let's look at the next part of the question. Figure 2.2 shows the pH decrease steeply and then after 18 hours remains constant. Calculate the time taken for the pH to decrease from 6.6 to 6.3. So I have just drawn that on the graph. As you can see it here, this is 6.6 and this is around about 6.3 here, this point, and then 6.6 was at 2 hours and 6.3 was at 6 hours. So 2 minus 6 is 4, 6 minus 2 is 4 hours, and that is what we had to give in the question. So you had to figure this out from the graph, and this was easy to do from the graph, wasn't something very difficult. This was 6.6 and you see it on the x-axis, and then you had to see it 6.3, and then you had to see that at the x-axis, and then you got to see that 2 6, so 1 was 2 and this was 6 hours, and the 6 minus 2 is 4 hours. Explain the results shown in figure 2.2. Now that was a very explain means you have to give me a lot of biology. There's no describe is no biology. So like you can see, you can understand why. You can see what the answer was. Lipase action produces fatty acids. Lipase converts lipids into fatty acids and glycerol. Fatty acids are acids and they decrease the pH. So decrease in pH. So lipase action produces fatty acids. Decrease in pH, steepest rate of decrease at start because substrate concentration was the highest. There was the steepest rate of decrease. Rate of decrease. Rate of decrease, very important that you write that. Then the rate of pH decrease slows because enzyme begins to denature because now the pH is changing. Then it plateaus at 18 hours at pH 6.1, enzymes are denatured. Optimum pH for lipase activity is pH 8 at the start. So this was the explanation that was expected, that you should give me this explanation. And the last part of the question was the scientists repeated the experiment using a higher concentration of lipase. Maybe they used 5%, now they're using 10% of lipase. All other variables remained constant. So substrate, temperature, everything remained constant. Predict how an increase in the concentration of lipase. So what did we have? We had, say, this is the lipase. So we had 100 first. Now we have 1,000 this time. So more active sites. The reaction is going to be faster. We have the same substrate, so it is going to be quicker. So decrease in pH would be more rapid because it said predict how it would affect the results of the experiment. Now this is where you would go wrong. This is where you would go wrong. And the results of the experiment. So decrease in pH would be more rapid. Plateau would be reached earlier, and plateau would be at the same pH as the previous experiment. So naturally, if you have, first you had 100 active sites and now you have 1,000 active sites, so everything's going to be very fast. Because first you had 100 active sites, so what did they do? Higher concentration of lipase. So you need to think in your mind, okay, we had about 100 active sites first, now we have 1,000 or 10,000. They didn't say how much, but they said a higher concentration of lipase. So this is how you think, and this is how you read the question, and this is how you interpret the question and then answer the question. Then we come to question 3. The circulatory system of mammals is a double circulation. Explain what is meant by the term double circulation. 1 mark. Blood travels to the heart twice for one complete circuit, or you could have said pulmonary and systemic circulation, and you could have given a little more about it, but basically you had to get your 1 mark. Then figure 3.1 is a photograph showing one valve in the mammalian heart, and you see these thread-like structures. And of course, you know what it is. It's you could have said tendons, you could have even said heart strings. Identify the structure labeled in A, figure 3.1, and describe their role during the cardiac cycle. So structure A is chordae tendineae, or you could have said tendons as well. That was also allowed. So you could get away with this tendon, not the correct labeling, which is chordae tendineae. Then valves are held in position. They connect the papillary muscle to the valve, prevent the backflow of blood to the atrium, and tension in the chordae tendineae during ventricular contraction. You see, when the ventricle contracts, blood could go back into the atrium. We don't want that to happen. So this is why tension in the chordae tendineae during ventricular contraction. The B part of the question was the endocardium is a thin layer of tissue lining the chambers of the heart. You remember the lining of the blood vessels is called endothelium. So the endocardium is a layer of tissue. It's tissue, not cells, and that's why we call those squamous epithelial cells. The tissue is called endothelium. So the endocardium is a thin layer of tissue lining the chambers of the heart. A serious condition called endocarditis results if bacteria infect this tissue. Endocarditis is treated with a combination of antibiotics. This increases the effectiveness of the treatment and reduces the risk of antibiotic resistance in bacteria. Table 3.1 shows the action of two antibiotics used together to treat endocarditis. Table 3.1, antibiotics used in the treatment, number 1 is gentamicin and number 2 is penicillin G. Now, gentamicin binds permanently to the bacterial ribosomes, and penicillin G inhibits an enzyme involved in cell wall synthesis of the bacteria. So they've given you two antibiotics and they've told you how do they kill bacteria by either binding to the bacterial ribosome, so that means the bacteria cannot make any more of their proteins, and the other is involved in cell wall. So no cell wall, so how will binary fission take place and cell wall be made? So that is going to be the problem, and the bacteria is going to die. So that's what antibiotics do, they kill bacteria. With reference to table 3.1, explain why treating endocarditis with a combination of gentamicin and penicillin G reduces the risk. So treating with a combination of gentamicin and penicillin G reduces the risk of developing antibiotic resistance. Now basically what you've got to understand from the question is you're using two antibiotics, so it must be something beneficial. Both antibiotics make sure all the bacteria are killed. Then if resistance against one, the other bacteria will still be effective. Then antibiotics have different sites of action. That's what's given in the question. And you would need two mutations to be resistant to both the antibiotics. So the bacteria would have to mutate, and there'll have to be two, because if one mutates and it's not being killed by one of the antibiotics, the other will still work. So that is why many a times doctors give two antibiotics during a very severe bacterial infection. And in tuberculosis we give three antibiotics. Then it says describe how the bacteria that causes endocarditis could become resistant to gentamicin. So part 2 of the question was a little critical. It says describe how the bacteria that cause endo become resistant to gentamicin. Course of antibiotic not completed, number 1. Not all bacteria destroyed. Random mutation of allele for resistance to gentamicin. So you see, mutation causes them to become resistant. So you've got to be very clear on this. How do they become resistant? So they become resistant like they will start to produce penicillinase. So penicillin antibiotic will not work on them. They will start to make gentamicinase or something like that, which will kill that antibiotic or that chemical which is being used as an antibiotic. Now, vertical and horizontal transmission. Now, this is important that you all revise vertical and horizontal transmission. So, an allele for resistance to gentamicin. I will just probably revise it at the end of this video. Next time antibiotics given, this will be selection pressure. Resistant bacteria will be selected for and susceptible will be selected against. Naturally, if you keep on giving this antibiotic, then the ones which are resistant will survive and they will become more. They will become more and more and more because they will not be destroyed and they will increase in numbers, and we'll say allele frequency changes. Next we come to question 4. Figure 4.1 is a scanning electron micrograph of a section of a plant cell wall. In living plant tissue, cytoplasmic strands form part of structure W. And you all know what are those cytoplasmic strands are the plasmodesmata, which are the connections between two cells. So they've given you structure W, and then of course they've asked you what that structure is. So identify the structure labeled W as plasmodesmata. Then describe the function of structure W, connecting two cells and movement of substance between the cells, and the simplest pathway, cytoplasm to cytoplasm, is the simplest pathway and a passage of cell signaling molecules. So number one, connecting two cells. Number two, movement of substance between cells. Number three, Simplast pathway and passage of cell signaling molecule. But you could only have written two and you could have got your 2 out of 2. So it wasn't something that you had to give me all these points. Then it said viruses can infect plant cells, outline the key structural features of a virus. Now that's something of the first chapter. Protein code, it has a DNA or an RNA strand, so that's 1 mark. Protein code, DNA or RNA, and some viruses have a phospholipid envelope or a lipid envelope outside. So 2 marks, 3 mark scheme points, any two, and you got your 2 out of 2. The next part is part 2. Many plant viruses can travel through the plant to enter companion cells in the phloem tissue. The viruses then travel with assimilates. Sucrose amino acids are assimilates in the phloem sap to other areas of the plant. Explain the mechanism that allows assimilates and viruses to travel through phloem sieve tubes to other areas of the plant. Basically, he wants to ask about the how the phloem, what is the way mechanism by phloem travels. So explain the mechanism that allow assimilates and viruses to travel. So virus enter via plasmodesmata, movement of assimilates into sieve tubes lowers the water potential. You see, sucrose enters the sieve tubes, lowers the water potential, too much of solute. Water enters by osmosis, hydrostatic pressure increases. We've done this in a number of questions. Unloading at sink lowers the hydrostatic pressure at the sink, higher pressure at source, lower pressure in the sink, so mass flow from source to sink, and viruses travel through the seed pores. So basically, this has come in. I mean, if I always ask you when you're revising a chapter, just do all the questions from that chapter, and you will see whenever we revise transport in plants, this is one of the favorite questions. And I always ask you to write it, take a picture, and keep it as a screensaver so that you can then memorize it for 2 3 days. And I usually do that. I make you do that in class, and then I ask you to take a picture and keep it as a screensaver. And of course, some of you do it to please me, and others, of course, will change the screensaver as soon as the class is over. But I'm okay with that. Now, just a quick revision of vertical transmission and horizontal transmission. Now, vertical transmission is this is the resistant parent cell, bacterial chromosome and plasmid replicate. Cell division takes place with this normal binary fission. Daughter cells each receive a copy of the plasmid and are resistant. So, resistant parent cell. Now, second is horizontal transmission. Now, this is a resistant cell. Say this is one of the resistant bacteria. This is the resistant bacteria. Say this is A. And this is some other bacteria, which is some bacteria B. Maybe it's a yogurt bacteria. Maybe it's a nitrogen nitrifying bacteria. Maybe it's some other bacteria, but it's non-resistant. Now, you see what happens. It's a conjugating bacteria. They form this tube, which is called a conjugating tube. And this plasmid, which confers resistant on it, is now transferred to this bacteria, which is the yogurt bacteria, or to bacteria number B. So a single DNA strand of the plasmid is transferred. Each bacteria then synthesizes a complementary strand, and both cells are now resistant. So now first A was only resistant. This was resistant. Now even B is resistant because it just forms a connection, which is called a conjugating tube, and the plasmid is transferred from one to the other. So the plasmid now goes into bacteria B, and now the bacteria B is also resistant. So this is called horizontal transmission. And this was called, and you can see why it is called vertical transmission. And horizontal means another bacteria just comes and shakes his hand and say, okay, hello, and he passes the plasmid to the next bacteria. So they're not related to each other. This, of course, is the parent cell, and these are the daughter cells. Well, I don't think we say that in bacteria, but so this is the original parent cell, divide, cell division, binary fission, and now we've got two bacteria, and they are both resistant. They both contain the plasmid. So this is vertical transmission, and this is horizontal transmission when two unrelated bacteria just come together, have a little handshake, and the plasmid is transferred to the other bacteria, and that's called a horizontal transmission. So just a quick revision of vertical and horizontal transmission. And with that, we'll end this video here. Thank you very much and all the very best.